So now I would like to introduce my idea. I've had this idea for a while now, and I actually want to bring it on and pursue my PhD in this topic. I think it's doable. I have a background in epidemiology from Columbia University, and I believe that technically and theoretically, this is really possible. I do. I think it's truly possible. I just think we need a proof of concept that it can work. And my idea is Wildtype.AI. If you actually go to the site, you can actually click on it. I've already registered the site, and you can see this. This is actually an image of the landing page. There's a link to my Twitter and my LinkedIn on this page. But let me introduce it. So introducing Wildtype.AI, the world's first fully AI synthesized human populations for pharmaceutical clinical trials. And I foresee Wildtype.AI launching services in a series of 4 phases. Phase 1 is AI synthesized placebo control groups. Phase 2 is AI synthesized active comparator controls. Phase 3 would be AI synthesized human subjects. Phase 4 would be AI directed individualized drug development. So here, let's dive deeper into the product. So the first product we intend to launch is an AI synthesized placebo control group. Drug developers will be able to create their own active treatment groups and compare that data to their own to our control group. This will cut the cost of placebo-controlled trials in half and increase the speed of clinical trials, since the sponsor only has to recruit half of the subjects. So how will we do this? We will systematically collect the vast abundance of real-world data from numerous sources. This will include data generated from placebo-control groups used in all previous clinical trials. It will also include scientific and clinical knowledge on the natural history of disease, patient data from electronic medical records, and data from observational epidemiological studies, such as the Framingham Heart Study and the Nurses Health Study. Once the data is collected, it will be analyzed, and AI subjects will be generated, which are representative of the subjects in the general population. The drug developers, they develop their own selection criteria for the study, and we generate an AI study population which matches these custom attributes. In cases where there are gaps in the data, smaller placebo-controlled trials would be conducted to fill in these gaps. And to account for a placebo effect in the treatment group, the bias will be controlled for with both treatment and placebo groups using AI technology licensed from other sources that currently already exists. So over time, the company will actively promote disease-specific universal standards for selection criteria in clinical trials, and we will promote universal standards for data collection and records for both clinical trials and electronic medical records to ensure uniformity and ease of data sharing and analysis. And early on, we acknowledged there may be some reluctance to trust the validity of data generated from AI synthesized control groups by both the industry and scientific community and health regulatory authorities. Because of this, for each study, the study participants will initially act as their own control group to validate the AI control group selected for the study. In instances where this isn't feasible, a small traditional control group will be used to validate the AI control group throughout the course of the study. So what we're looking here is that I truly believe we have such an abundance of data now, and the analytical tools possible where we can really take and simulate control groups. I really do. I really don't believe from a scientific perspective there's a need for if you're testing a drug for depression to have a new placebo group for every new drug testing for depression. I understand that there'll be differences in selection criteria, and that's why we discussed overcoming those challenges. But I really think by truly understanding the natural history of disease, truly understanding placebo response, and having the abundance of data that's already been generated, we could actually create a custom group with existing data for future drugs going down the line when it comes to placebo controls. And the second product in my proposal is an AI synthesized active comparator group. And an AI control group with an active comparator will be developed for each and every drug on the market. We will collect data generated from market approval of drugs on the market. We will combine this with data generated from post-marketing phase 4 studies and registry studies. Finally, we'll collect data from electronic medical records and all those studies which generated data on the specific therapeutic. As with AI placebo-controlled products, we'll generate a custom AI active comparator control group based on custom design of the drug developer, matching the group to the selection criteria and validating the control group to ensure validity of the data generated. And this goes back to the previous idea again, is that if we're comparing a new asthma medication against current products, we know how the current products work. They've been tested. They've been tested in clinical trials when they were being approved. They've been tested by doctors, and that data has been recorded in medical records. There's no reason to have a new active comparator group developed, paid for, which ultimately just slows down a clinical trial. There's no reason to create that from scratch for every new asthma drug that comes out or any other drug. Now the logic is that you're introducing bias. The same variables and factors, environmental factors, social factors, economic factors that played a role in the first trial that could be balanced for through randomization. They're different. They're different with this trial. And I agree. I 100% agree with that. That's like, year one of grad school for epidemiology, that you understand that concept. So, but I think with modern AI, modern advanced data analytics, we can control for those factors, especially when we don't just have one study, we don't have two, but when you're combining it to EMR records and vast amounts of data, you can control those out. You can make what what you truly should expect from somebody receiving an active comparator, and you can match it to the study. Now, my idea is like, and if people have questions about that, then let's use the subject as their own control at the beginning. Take it and compare it to the data we generate with these AI synthesized active comparator controls. And if they match, if the bell curves overlap each other perfectly, great. That's what we're expecting. And these concepts could really be validated by doing them in parallel to a current clinical trial. And this is what I would love to be able to demonstrate and prove in a PhD program and in future research throughout the course of my life. I would love to do this. So, phase 3, let's skip ahead to the year 2030. Wildtype.AI is the world leader in AI clinical trials for drug development. We've collected and generated data that dwarfs the entire internet a decade prior, and we have server farms in rural Utah that rival those held by the NSA. The royal gem of the company is the world's strongest IBM quantum computer, custom designed for pharmaceutical discovery, research, and development. In development of our third product, AI synthetic human subjects, the company has hired the brightest minds in science and has cataloged every protein and cell receptor in the human body. And we have mapped the cascading interactions with each after exposure to every drug and toxin known to man. Our quantum computers are able to synthesize the full spectrum of pharmacokinetic and pharmacodynamic data for every new drug being developed. The idea of first-in-human trials being dangerous is a thing of the past. Drug developers can enter clinical trials knowing their drugs are safe and having a relative understanding of the expected efficacy of each compound. Clinical trials in humans are small and are simply used to confirm the knowledge generated by AI. The average time to market for a drug is a single year. when we have a compound, we have that variable. We know, especially with small molecules, we know how it's shaped. We can trace it through the body. And if a molecule comes by that's slightly different, it generally, for the most part, I mean, there's exceptions where things don't go as planned, but generally, we can modify a molecule slightly and make a slightly different drug, but it has the same effect. We see this with amphetamine salts. You slightly alter them slightly, and they have an effect in some and not others. They have more beneficial effects in some populations, but we can tweak medications that way. Well, if you do that enough times, and you feed that data into AI systems, you get to the point that you can actually start to predict, what the effects will be on different on the biochemistry of the body. Well, what will happen to different cell receptors? What will the cascading actions be? And if you into AI systems, if you keep feeding that data more and more, it gets to a point. Especially when proteomics and genomics starts to get, it gets more advanced, and we have a better understanding of the human body, the AI should be able to track what happens down each cascade and its effects on the body. You should be able to predict new compounds, what will happen throughout the body. Now, where quantum computing comes into play is that quantum computing is able to make simultaneous calculations, millions of them simultaneously, instantaneously, and their interactions with each other. It really changes the way we can actually analyze how, millions of proteins in the body can suddenly react from one molecule entering it. I think that will be the key to making this thing possible. And I chose 2030. I truly believe by 2030, the technology, whether it's being applied or not, the technology will exist, that this could be a reality if we get a jump start on it. And I think the previous two phases that I proposed would be the precursor to actually making what you see now on the screen truly a reality. So let's go on to the next phase, our fourth and final, our final product, our magnum opus, our piece de resistance, is AI synthesized drugs individualized for each patient. The product analyzes the complete genome and proteome of each patient, which is combined with previously developed knowledge and technology and custom designs, develops and manufactures a drug solely for that one patient and their health condition. The clinical trial industry has faded into history. Humanity and the world, are a different place. This is, I think, the preceding three programs I proposed. I really think this is where it would lead to a one patient, one drug market, truly individualized medicine that truly treats the disease specific to that individual's their environmental, social, economic, physiological factors, everything taken into account for. And we truly treat patients on a one-to-one basis. I think this is the future given this path, and I think this is a reality. And once this comes about, I think the pharmaceutical drug industry will be there. It really will, but it will be a completely different place. I think medicine will be a completely different place by this time. And this is the vision I have for the future. And I gave my references for everything I presented, and I thank you for your time. And thank you. Have a great holiday. It was great being with all of you in the class, and I hope to study or work with you in the future. Thank you. Bye-bye.